Thank you. And today I will talk about implantation and endometrial disorders. Then I will go through the two main objectives of our project, and I will conclude with the future perspectives. Whoa. So what is embryo implantation? Embryo implantation is when the embryo or the blastocyst is attached to the epithelial layer of the endometrium. It can only happen when we have, It can only happen when we have a receptive endometrium and a competent embryo. These happen in the mid-secretory phase of the menstrual cycle, and it normally occurs 6-10 days after ovulation. In this window of implantation, the two main actors are the progesterone and the estrogen. During this period, we have also a remodeling of the endometrium and a lot of changing happening, like the decidualization, where we have the stromal cells that can differentiate into epithelioid secretory cells. There are a lot of things that can go wrong in this implantation, and this was already studied in animal models principally, and this is just to say that the human reproduction is very low compared to the animals. There are a lot of reasons behind this, and it can be genetic causes or gynecological disorders. The one that we are interested more are endometriosis, fibromatosis, and adenomyosis. All of these conditions have in common the fact that they have problem in the implantation process, and they have a disruption in the vascularization, so what it concerns the endothelial part of the endometrium. To study and to decipher the window of implantation, we were using animal models, and, but as everyone knows, the animal models, especially in the field of reproduction, they do not resemble the human physiology because they have different menstrual cycles, and they have, of course, high cost and a lot of ethic concerns behind. So for that, we are substituting these models also to be in line with the 3 R's of animal research: reduce, refine, and replace. We are substituting with the 2D models, they are very simple for the setting any experiment, but then we want something more complex, like the 3D models. And now we are also looking at organ-on-chip models, where we have also the diffusion of the nutrients, like it happens in the human body. Whatever model you choose, you can generate it using immortalized cell lines and primary cells. These primary cells can be retrieved from endometrial biopsy or menstrual blood. That is our new source, because it's a less invasive source for these primary cells. So the two main objectives that we are pursuing is, one is to establish a reliable and non-invasive human endometrium 3D model in a static condition that is primary cells based. And the second one, that is a donor-independent model, is an organ-on-chip model, so in a dynamic condition. The first part of the first objective was to recruit donors and give them the instruction to the use of the menstrual cup. The second part was to generate the model and then stimulate this model using the hormones that are the actors of this implantation, so the estrogen, the progesterone, and the cyclic AMP. Also, we wanted to see if this secretory phase that we are mimicking is possible to be reversed using the mifepristone. Is a drug that is an antagonist of the progesterone. And then we analyzed our results through immunofluorescence, qPCR, and scanning electron microscopy. The first part of the result was to see how phenotypically looked our menstrual blood-derived organoids compared to the biopsy one that are very common in our field. So we didn't see any significant difference in the phenotype of these models. And we wanted also to look at the composition, the cell composition of our model. So we looked at the marker of the stromal cells that you can see here in red, and the marker in green that you cannot see really very well in this slide, but there is. In green is the epithelial cell marker. And this is not common in our field because everyone says that organoids are only made by epithelial cells. But no. Same was for the biopsy counterpart, as you can see in these two pictures. Moving on, the organoids also have a biomarker for the implantation, and we wanted to check if after simulation these organoids could express the marker, that one of the markers for the implantation, that is the glycodelin. As you can see in both derived organoids, we can see in green the expression increased in the simulated organoids. We wanted also to verify the gene expression of the markers, if we could find in vitro this expression, and we were happy to find that IGF-PP1, that are the most famous decidualization marker, prolactin and ZBTB116, they are significantly upregulated when we added the hormone cocktail, and they significantly downregulated when we added the progesterone antagonist. Moving on, we wanted to see if the ultrastructure of this model could be the same as the epithelial layer, because we know that epithelial layer, to attract blastocysts, can develop these pinopods that are this structure that you can see in this picture. Was already demonstrated for biopsy-derived organoid, but we wanted also to show that our menstrual blood-derived organoid can develop this feature that is very important during the implantation. Moving to the second part of our project is to develop an endometrium on a chip model. We were using the tissue chip system. Is a controller that is connected to with the micropumps to this chip. We used two types of chips, the PDMS and the COC. And this chip is formed by two cell culture compartments where we put our 3D model, and these microfluidic channels that were endothelialized. So we added the endothelial counterpart in these channels. So the experiment starts with seeding the endothelial cells in these microfluidic channels. After a few days that the cells were growing, we added the 3D model formed by stromal and epithelial cells, of course cell lines, because we wanted a donor-independent model. Then we started our treatment using, similarly with the organoids, nothing for the vehicle, the hormone cocktail to mimic the wind of implantation, and the hormone cocktail plus lipristal, another progesterone inhibitor. And then we analyzed our results with analyzing metabolic profile, the cell composition, and decidualization markers. Okay. The most important part of this model is that we added another compartment, that is the endothelial part, that is very important in many pathologies that I already mentioned before. The cell types that we used, I will go very fast on this, because are endothelial cells. Those are primary cells that we use to endothelialize the channels. And these two cell lines, the epithelial EM42 cell lines and the stromal cell line, in a ratio of 1 to 10. The first part of the result was on the endothelial part, so we wanted to see if these cells were alive in the channel after 10 days of culture, and we saw that this is a calcine AM, is a marker of live cells. So we saw that after 10 days, these cells were still alive in our chips. Okay, so in here we checked if our marker for endothelial cells, VE cadherin, was present in our experiment, and we wanted to see if these cells were looking as a vessel in our system. So then we did this metabolic analysis to see if the cells were in good health in our system. So we used the LDH marker for health of cells. And we saw that, of course, the level increased after 10 days, but it was still normal levels. Then we analyzed through digital droplet PCR the results for the main marker of receptivity. And we analyzed IGF-PP1 the same as we did with the organoids, and we saw that these markers were all expressed in these simulated 3D models. The same at the secretion level, when we analyzed the IGF-PP1 secretion protein level in the channels, and we saw that in both type of chips, upon the treatment, we have an increase in the secretion in the serum of this IGF-PP1, and after the stimulation with ulipristal that serves to block the effect of the cocktail, the hormone cocktail, we have a significant decrease of the IGF-PP1. Then we characterize our 3D model made by stromal and epithelial cells. And we were surprised because we bought this cell line, EM42, and should be epithelial cell line, but we analyzed the marker of stromal cells, that is vimentin and cytokeratin A18 for the epithelial part. And we saw that this was a mixed cell line. Then we used this stromal cell line, and we mixed to make the 3D model. And this was the droplet that we generated. So also for the whole experiment, so with endothelial and 3D model, we analyzed the level at the metabolic profile, so always glucose, lactate, and LDH, and we see the increase in the LDH level. This can be improved in further experiments. Okay, and we also checked all the biomarkers that are used for the window of implantation. And as you can see, all these biomarkers in the condition of hormone-stimulated cells was upregulated in both type of chip and also in static condition. And we see a downregulation when we added the ulipristal. The same was for the secreted protein that we were analyzing, IGF-PP1 and the prolactin. So we found that for these two main that were not studied before, FKBP5 and OXA10, are significantly increased and can be used as biomarker for the endothelial part in our models. And what we are doing now is the expression, the analysis of the expression in this organ-on-chip model of the metalloproteinases that are very important in physiological process and in pathological process that we are studying. So for now, I just have a preliminary result on some metalloproteinases that are more expressed in the dynamic condition and other inhibitors of these metalloproteinases. Concluding, I think we reached the goal to have these reliable models in one that can be used as personalized medicine and one that can be used as a donor-independent model for many diseases. What we are looking now is also the expression that can be carried or what can be important on the exosome level in our 3D model, the exosome that are released in our model, and the proteomic that we are performing to understand what are the pathways involved in many implantation failure. So I want to thank my mentor Paola Piomboni and all the lab that is participating with me in this project.